Hey guys, so welcome back. We are here in our next section all about genetics. Yes, this is a longer set of notes, but this will be the only notes that we take for this week. And we'll be moving through pretty quickly now. Again, like we covered the topics before we left for break, and they were very difficult topics. We're going to slowly start to ease back into it, and things are going to become easier and easier as we go through the rest of the year. So here we are talking about Mendel and heredity, so please label this section 6.3 and 6.5, Mendel and heredity. We've got three key concepts. We have Mendel's research showed that traits are inherited as discrete units. Genes encode proteins that produce a diverse range of traits, and the inheritance of traits follows the rules of probability. We're going to talk about all those things in detail. Vocabulary, there's a lot of it, and again, we're going to talk about all these things as we move through. We've got heredity, traits, genetics, genes, law of segregation, alleles, law of independent assortment, cross, dominant, recessive, homozygous, heterozygous, genome, genotype, phenotype, purebred, Punnett square, monohybrid cross, and dihybrid cross, and then lastly, the word probability. Two essential questions only. Where does an organism get its unique characteristics? And then how can we use probability to predict traits? So yes, there's a lot of vocabulary associated with the section, so memorizing some of the vocabulary might be difficult, but when you get down to the gist of it, our essential questions, it's not as difficult of a section as you actually think. So Mendel, who was Gregor Mendel? Well, Gregor Mendel was a monk who lived in the 1800s in Austria. And if you remember, I talked about monks before. Monks were very learned people. They studied in monasteries, and there were a lot of books, a lot of riches, a lot of rich families sent their sons to the monasteries to learn. So Gregor Mendel was a monk, and he is the first person to develop rules to accurately predict the patterns of heredity. And he did this using pea plants, because at the time, pea plants were very easy to grow and inexpensive. They grew very fast, and you could easily observe some characteristics. So the modern genetics that we actually use today are based on Mendel's explanations for patterns of heredity using these garden pea plants. All right, so what exactly is genetics? Well, the delivery of characteristics from parents to offspring is called heredity. You know this word. The scientific study of heredity is called genetics. And in genetics, we study physical features, and those physical features that get inherited from your parents are called traits. So traits are specific characteristics such as like the color of your hair, the color of your eyes, how tall you are. If we're talking about pea plants, we're talking about what the seed color. Is it yellow or is it green? We're talking about plants themselves. How tall is the plant? So trait is just a specific characteristics, and those are all controlled by genes. So Mendel found that traits are controlled by factors that pass from parent to offspring. And again, you know this word already. We call them genes. And genes are the portions of DNA that determine our traits. Now, when we talked about this last, we talked about how as human beings and most organisms that reproduce sexually inherit two copies of each gene, one from mom, one from dad. So organisms donate one copy of each gene in their gametes. So the sperm donates one copy, the egg donates one copy. Thus, the two copies of genes are separated when these gametes are formed, right? Because you have two initially, they separate through meiosis, right? And then when you go on to inherit your traits, you get one from mom, one from dad. Because those genes separate when the gametes are formed, the sperm and egg are formed, we call it the law of segregation. This increases our genetic diversity. Now, you get two copies of each gene, and those different forms of a gene are called alleles. We'll talk about that more in depth. So here's just this thing about independent assortment. So the law of independent assortment states that the allele pairs separate independently of each other during the formation of gametes. And again, the gametes are the sperm and the egg. We talked about meiosis back before break. So in forming a sperm or forming an egg, those cells divide, and then they divide again, splitting that chromosome number in half. So you start with 46 chromosomes. At the end, you end up with 23 chromosomes in each of the sperm and the egg. So that when those sperm and the eggs unite to form the new organism, they have the proper amount of genes, their chromosomes. All right. So Mendel's experiment, what did Mendel actually do? Well, he crossed several different types of pea plants, okay? And to cross means to mate or breed two individuals. So what exactly did he do? You don't need to write this down. This is a lot of extra details. But basically, in order to breed those two plants together, he took the male parts and removed them from the first flower to make sure the plants would not self-pollinate. And then he'd take pollen from the male parts of the second flower and kind of brush it onto the female parts of the first flower. So he could know which two he were actually crossing and breeding together. Why did he study pea plants? Well, they have very easily observable characteristics. This is important for you to know because on the EOC they might ask you, why did Mendel study pea plants? Well, as pea plants, they were cheap, they grew really, really fast, and they have all of these different characteristics that are contrasting. You can tell the difference immediately, like the flower color. Some plants have purple flowers, some of them have white flowers. You can easily tell the difference. The seed color, some have yellow, some have green. The seed shape, some are really round and some are kind of like wrinkled up. The pod color, the pod shape, and then also flower position. Is it in the middle of the stem or is it at the end of the stem? And then lastly, plant height is another one. Were they tall plants or short plants? So he could easily observe these plants and breed them together to see what the outcome was. Now, Mendel developed several hypotheses to explain the results of his experiments. And his hypotheses are basically correct, but they've been updated at this point with more like newer terms and more complete knowledge as we learn more about genes and stuff like that. But the very first really important thing that he discovered is this thing that he called the principle of dominance. And it states that some alleles are dominant and others are recessive. Okay, so the different forms of gene. There's one that's going to be dominant and there's going to be one that's recessive. The recessive alleles, the recessive versions of the gene, are only expressed when the dominant allele is not present. And we'll go on to explain these, so just write the vocab now. And as we go through this week, we're going to talk about these and we're going to use these words more in context so they'll make more sense to you. Now, although Mendel didn't use the term allele, and this part at the top you don't need to copy, he used a code of letters to represent the function of alleles, so dominant versus recessive. The dominant letters, the dominant alleles, are always shown with a capital letter, okay? And it's usually the letter from the first letter of the word for whatever trait it is. So if we use purple flowers being dominant, we use a capital P to represent that purple color. Because it's dominant, we're going to use a P, capital, purple, P. Now, recessive alleles are shown always with a lowercase letter, and the letter is always the same as the one that's used for the dominant allele. So if we're saying that purple's dominant, now we're going to say that we also have white flowers. White is recessive. We're not going to use a little W, we're going to use a little P to represent it because our dominant color is purple. We've already dictated that we're going to use P to represent that, and it's going to be capital P because it's dominant. So the recessive color, whatever letter is the opposite, we're going to use the little p. Okay. So we have these other terms, homozygous and heterozygous. So if you have two of the same alleles of the gene, we call you homozygous. Homo just means same. So a plant that has two white alleles, two white colored genes, are going to be little p, little p. So that would be our flower color because it's recessive, it's going to be white. If an individual has two different versions of the allele, then we call them heterozygous. Hetero just means different. So if they're heterozygous, they're going to have a big P and a little p. Their color is actually going to be purple because the big P overshadows the little p. So combinations of the alleles determine our traits. So all the organism's genetic material is called the genome. And the set of alleles that an individual has for characteristics is called the genotype. So whatever your genes are your genotype. The physical traits that you portray are your phenotype. You can remember this like genotype, what genes do you have? What letters? Do you have a big P and a little p? Do you have whatever letters they are? That is your genotype. Your phenotype is what you physically look like. Are you purple or are you white? And our genotype, again, determines our phenotype. So, for example, if we have the genotype of little p, the phenotype of the plant is white flowers. If the genotype is big P, the phenotype or the physical characteristic is that it is purple. Okay, if you have a big P and a little p, guess what? That big P is dominant, so it basically blocks out that little p, so the flower would be purple. So Mendel wanted to see what would happen when we crossed individuals that have differing traits. So he started with plants that were homozygous for his traits, and he called them purebreds because they had the same genes, okay? And they're the parent generation. As he moved through the next couple of generations, I'm going to go ahead and hit that. So he crossed parents that had different traits, and that resulted in offspring called hybrids because they had a combination. They had a big P and a little p. The first ones, he knew what they were. Like little p's and little p's would always have little p's and little p's. Okay, but if you cross something that has two different ones, okay, they're heterozygous, you're going to end up with different qualities. The first generation of hybrids he called the F1 generation. And then if you took two of those ones from the F1 generation and cross them together, you got the F2 generation. Take two from the F2 and cross those, you get F3. Take two from there, you get F4, and so on. Okay, so what do you notice? If he took the purebred purple flowers and the purebred white flowers and crossed them together, he'd end up with all purple flowers. But then if you take these ones and you cross them together, you would end up with some that were purple and a couple that were white. And here's the same picture. So again, here's our genes, big P, purple flower, little p, white flower. Cross them together, we end up with these genes, big P, little p, big P, little p, big P, little p, big P, little p. Okay, all of these are going to be purple because they have a big P. Now, if you take two of these and cross them together, okay, you end up with some that have big P, some that have big P and little p, and then some that are just little p, little p. And we're going to talk about that more in depth when we talk about things called Punnett squares later this week. So Punnett square is a model that predicts the likely outcomes of a genetic cross. And it shows all of the possible genotypes that could result from that cross. And we use these to make very simple predictions, okay? They're not 100% certain, but it shows a possible outcomes, okay? So we can calculate a probability based on these Punnett squares. And again, they're for very, very, very simple traits. Probability is a likelihood that a specific event will occur. And you use this a lot in math, so you're very familiar with this term. Okay, so for example, this is how you would use a Punnett square. So we have this little blank, and we want to figure out how many outfits can we make based on the clothes that we have in our closet. So if you look at here, we've got, for shirts, we've got a red shirt, a green shirt, and a yellow shirt. Okay, we've got blue shorts and blue jeans. Now we want to figure it out. So we could put a red shirt with our blue jeans to make one outfit, our green shirt with our blue jeans to make one outfit, and our yellow shirt with our blue jeans to make one outfit. Then we could take our red shirt and our shorts to make one outfit, our green shirt and our shorts to make another outfit, and our yellow shirt and our shorts to make another outfit. You can see with my hands, I'm kind of bringing one down and one across to meet it. Same thing, one down, one across to meet it, one down, one across to meet it, so that I'm filling in those blanks using that little chart. That's exactly how you fill in a Punnett square. I'm going to show you a video after we take our notes so that it's much more familiar to you on how to actually fill these in. So there are two kinds of crosses you can do. You have a thing called a monohybrid cross that involves the inheritance of one trait, and then a dihybrid cross, which is a little bit more complex and involves two traits. Now, dihybrid crosses with heterozygous alleles, two different versions, if we cross parents that have two different versions, will always have a ratio of 9 to 3 to 3 to 1. And again, we'll talk about this a little bit more in depth this week and at the beginning of next week. And here is some more little Punnett squares. And again, if we take a homozygous cross, okay, parents have both dominant or recessive alleles. Okay, heterozygous is two different alleles, a big one and a little one on both of the parents. And again, we're going to watch a video next on going through how to actually do these Punnett squares and go more in detail with the vocabulary that you just took. So don't be stressed out if you aren't following me at all. You will get it, I promise you. All right, guys, have a great one.